All right, so now that you've done your blood spatter, now we're going to take a look, let that dry. While that's drying, we're going to take a closer look at DNA. So DNA, if you pull open that file folder with biology information, open that up and try to remember as much as you can about DNA. But DNA has several segments, and it's a very important molecule because it's something that gives us our traits. So it tells us that we're going to be this tall with this color eyes, this color hair, this shape lips, this size nose, hitchhiker's thumb or not, widow's peak or not, attached or detached earlobes. I mean, there's so many things that it gives us that information. And however our letters are arranged, and letters, we'll talk about those in a second, that's what gives us the traits that we have. And so very, very, very important. We're going to take a look at it here to be able to take a look at DNA and help us to be able to determine whose DNA we can find at the scene and see if that gives us any clues as to what caused Anna's death. So if you guys take a look back at your document, and mine's being kind of goofy. It's really in a goofy spot. So let me try to scooch it around a little bit so that you guys can access it a little bit better. Like I said, I don't know why it's being so silly, but hopefully you can see you guys a little bit better. What it says is this is the do-it-yourself DNA, and so this is the page that I want to make sure that you get completed, or at least you get to use the models to try to build this DNA so that it will make sense. Now I will tell you, just like some of the other pages, I also gave you some links to look at to help you if you're like, oh my goodness, I still don't remember this. If you go to page 7, you'll see that there's a couple links there. So please take a look and access that information. But as you read through the material, you're going to put some background info, and the more specific you can be, the better. And then you'll see there's this drawing, and so you're asked to pick different colors. Now, I will tell you, I would probably recommend doing this in your hard copy and then taking a picture of it and submitting it, so you don't have to worry about filling all the circles and coloring this drawing. But all of this should be colored to signify what all of these different components are. And that's going to also help you with the information on the next page. So just make sure that all of this makes sense. When you go to that last page then, the one that you need to do before you leave class today, you'll see there's all these pictures. And so you're going to use these pictures to fill in this information in this box down below. So this first row is just looking at figure 1. So you're going to determine with figure 1 which letter is pointing to each of these different components, and make sure that makes sense. Otherwise, you don't really understand that structure of DNA, and that's what this is asking us to do. Then when you look at the additional questions, and it's not really letting me scroll down at all here, but that's helping to have you look at these other figures, and it says put the appropriate figure number to be able to determine what's what. And so if we take a look at the pieces, you guys are going to work in your groups again, but there should be a baggie for each of you. And so in this baggie are all the different pieces that you need to be able to try to build a little section of DNA. And you're going to have one, and your partner, there should be enough for your partner to have one as well, but you guys can kind of play around with it and hopefully make some sense of it. So if you guys dump out your pieces, and I want you to kind of organize similar pieces together. So basically what you should see is you've got 4 pieces that are kind of bigger, and if you look, they have letters inside of this middle ring. This one says A, this one says T, and then there's 2 other ones as well that say C and G. Now those letters, A, C, T, and G, those are the 4 what we call nitrogenous bases. So that's this first question. It's asking you about the bases, and I shouldn't say that's the first question, I'm sorry, it's not the first question, but if you look over behind where my picture is here, you should be able to see where it's asking for a nitrogenous base, and that's what this is showing right here in figure 3. So you should put figure 3 as an example of a nitrogenous base, which is one of these. And how do you remember that? Well, they drew in the ends there so you can see where the nitrogens are present. So what you want to do is you want to take your nitrogenous bases, and we have to hook up deoxyribose, or the sugar. That's what deoxyribose is. So the sugar is what looks like this 5-ringed structure that's also got a red and a gray sticking off of it. And so what you're going to do, sorry, my lights went off, is you are going to carefully connect this crazy blue piece that kind of moves itself around into the silver. So there's basically an innie and an outie, and we've got to connect them, and they only go a certain way. So make sure that you're inserting it properly, but you should, you might have to press kind of hard, but that's what we want to do. And what you're going to do is you're going to do this with 2 of your pieces. So here's my recommendation, because they'll only go together this way. A always has to go with T, or T always goes with A, and then C always has to go with G, or G with C. And how we know that, I'm going to fast forward a little bit, but you'll see that they have magnets to bond them, and these are hydrogen bonds. But what happens is they stick together. But we can't put C or G here because C and G have 3 connectors, or 3 hydrogen bonds, whereas adenine and thymine only have 2. That's why they always have to connect that way. And the really cool thing about this in our body is this is why we're able to make exact copies of our DNA whenever we make new cells. So that way we make sure that all of our cells contain the same information that says, hey, I'm Mrs. Lindy's cell, or hey, it's your cell, whatever the case may be. So what happens is, I did A and T, you can do C and G, but you have to do them in those pairs. So you're going to have an extra set that won't have the deoxyribose or the phosphate. So deoxyribose connects right to the nitrogenous base, and then the next piece that we have is the phosphate. And so when we take a look at the phosphate, you should see that there is a red that's got an opening, and that's going to hook with this silver piece on the deoxyribose. And remember, when you look at your lab, so this is the deoxyribose, so that on your paper is 1, 2, 3, 4, figure 5, and then figure 4 right in front of it is the phosphate. So make sure you're filling in the charts accordingly. But you hook the phosphate, like I said, where this opening is on the red, you hook it to that little sticky outie on the gray with the deoxyribose, and you're gonna kind of have to push it hard. Well, you guys, mine's just not wanting to stick. And then you're gonna do that same thing with T. So you're going to put the deoxyribose with the nitrogenous base, and you got to move it so that it will connect properly. And then you're going to put the phosphate group onto you guys, I'm connecting them wrong, aren't I? That's why I'm having trouble. Sorry, you got to do it on this gray that's got this interesting little sticky outy piece there, right? That's where we want to put them. Sorry for my confusion. We'll try again. And so then, here comes the magic. Now we can stick them together with those hydrogen bonds. And so when you stick them appropriately, this is what happens. But these are the hydrogen bonds connecting the 2 nitrogenous bases. And then connected to those are the deoxyribose sugar and the phosphate groups. And what happens is they're going to basically line up like this, and then they can connect to more things. And what happens is you can build these together and keep on building them. And eventually it makes what DNA actually looks like, which we call a double helix, because it's this twisted ladder. So it looks like this twisted ladder staircase. It's going to spiral and spiral and spiral, but containing all of our genetic information. And I just got to show you the picture on the box so that you can see that's what it would look like if we had enough pieces to be able to put it all together. But it's just important for you to understand the components, what has to fit together to be able to create this, what we call macromolecule that's in our body that really does a lot, because not only does it give us directions for what we're going to have for genetic information, it also does something else very important. It tells our body the directions for how to make the proteins that our body needs. So really, really great stuff. And like I said, just trying to give you a little bit of a hands-on to kind of see what this looks like. Now I'm going to ask you guys to carefully take these pieces apart when you're done, and then you're going to put them back in the baggies, and then I will disinfect them when I return to class tomorrow. But if you could all put them all back up on that front table, that would be much appreciated, and put them away in the bags. I would greatly, greatly, greatly appreciate that. But hopefully this was enough information to get you what you need. Take a look at those other videos to fill it out as well. And then, like I said, if something doesn't make sense, please be sure to speak up. So now, hopefully you have about 10 minutes left in class. So head on back. Like I said, grab one of the clear rulers. Both you and your partner should have one of those clear rulers, and measure on the centimeter side. So every little dash is a millimeter. So measure how many millimeters each of those droplets were. And then you're going to record that information and put it in the graph. That's basically how you're recording it. And if you look, there's that video from Mrs. Hisrick that tells you this is how you do it. So take a look at that information. And like I said, if you do have any questions while you're taking a look at this, please don't hesitate to send me an email, and I'll answer you as best as I can. And then also make sure that you get a picture of your charts of your blood spatter before you leave today. And the other big thing that Heidi says as she goes through this is she puts little X's as she measures them. I had a group today when they were doing this, and they wrote on there how big that measurement was, and then they could just use that for all of their data to put it in the graph. I thought that was a great idea. But that's what you want to have all of this notebook completed for next time. There's additional information with the DNA information with the Canvas, not with Canvas, I'm sorry, but with the MyPLTW. So I was just going to show you this here. It talks more specifically about these different colors and what they represent. So definitely take a look at those particulars so you can, develop a little bit more understanding. But like I said, hopefully this gets you where you need to be to fill all this in and have it completed and turned in on Canvas by the next time we meet. So that would be October 7th. Enjoy the rest of your week, guys. I'm sorry I didn't get to see you in person, but have a great week, and we'll see you next week. Bye-bye.